Very good day, students. So today we are here for our very first topic in MSc semester 1 for the subject of functional organization of animals. So here I want to recall that we are starting with our unit second, which is the transport and circulation. So this is the very first topic in unit second, and this topic is the circulation among protozoans. Now, as we all know that protozoans have got their name because they are very small in size. So if we go by the first part of this word, which is proto, so proto means very small, they're small, and zones means animals. So these are very small microscopic animals which are found primarily in the fresh water. So we can see them in microscope, as we have seen amoeba, we can have paramecium, we can have euglena, and so on. So some protozoans, they are amoeboid like amoeba. Some protozoans, they have got cilia, so they are called as ciliates. We have got a group called ciliata, and then we have the third group, which is flagellates. So they are having flagella like euglena. So what is the topic containing? We have to be very careful about the transportation of the food materials and the gases in protozoans. So first of all, we'll be starting with the circulatory system. Now, what is the main function of circulatory system? The main function is to transport gases and to transport food materials. So this is the main function of the circulatory system. Now, if we talk about ourselves, we have got a definite particular circulatory system. We have a heart, we have arteries, we have got veins, we have got capillaries. So what is the main function of the blood? So it is to transport the food materials, and it is to transport the gases, which is oxygen and carbon dioxide in our case. So this is the primary function of any circulatory system. So this is to distribute food materials and to distribute the gases. The next thing which it will be helping in is the removal of waste. So this is again a very important you can say feature of circulatory system. So it is also removing waste, it is also carrying waste from one part of the body to the excretory organ or the kidney in our case. So this is again involved in the circulatory system. Now coming back to protozoans, as protozoans, they are unicellular organisms. So there is an intracellular fluid. So every protozoan has got intracellular fluid because it is a single cell. So there is no concept of intercellular fluid or extracellular fluid. So we have got an intracellular fluid in case of protozoans. Now, nutrients, they enter the body directly from the large extracellular environment in which they are living. Now suppose a protozoan, it is living in fresh water. So fresh water is devoid of any kind of salts. So fresh water has got very low salt concentration. So but it is containing food. So that means these protozoans, they are getting food from the fresh water in which they are living. Now what about the waste products? So they are again given out into the water. So main waste product in protozoans is ammonia, NH3. So it is directly given into the fresh water, and that is excreted out simply. So there is no role of excretory system as such in case of protozoans. If we talk about the gaseous exchange in case of protozoans, it is again simply by diffusion. So here what happens, that oxygen is more outside and it is coming inside the amoeba simply by diffusion, and when it is converted to carbon dioxide inside the amoeba, it is going out due to high concentration inside. So the gas exchange is also with the diffusion, so there is no respiratory system as such. So these points are very basic about this topic, and actually what we are studying is that if an amoeba is taking in food by just, suppose it is taking food by endocytosis. So if the food is coming inside amoeba, it is inside the food vacuole. Now how this food vacuole it will be transported inside the body of the amoeba is the circulation. So we are concerned with the transportation of food vacuoles along with the food. We are concerned with the transportation of contractile vacuoles, the formation of contractile vacuoles, the vesicles. Then we are concerned with the excretory processes, that how the excretion is regulated with the cytoplasmic movements. So this is the course of circulation in protozoans. Now first of all, I am taking the concept of cyclosis. So what is cyclosis? This is the movement of cytoplasm inside the body of the animal. So here the animal is a protozoan, so this is a single cell. So the movement of the cytoplasm within the cell is called as cyclosis. So there can be various factors upon which this cyclosis depends. So the speed of cyclosis or the direction of the cyclosis, it is again very important in case of protozoans. So we'll be discussing it later on. Now this cyclosis, it depends upon various factors. So first very important factor is the presence of contractile proteins. So these are basically the proteins which are very comparable to myosin and actin, which are present in the protozoans, and they are contractile proteins. And whenever these proteins they go for polymerization or depolymerization reactions, so that help in the cyclotic movements. So as we know that amoeba, it is moving with the help of two processes. First is gelation and second is solation. So gelation is the formation of gel and solation is the formation of sol. So these are reversible kind of reactions. So gel will be converted into sol, that will be solation, and sol will be converted into gel, that will be gelation. Now if we take, suppose we are taking one amoeba, suppose this is amoeba, and this is the nucleus. Now what happens that if this is the direction of the movement, if it is going towards the right side, so this will be the zone of gelation. So soul will be coming from this side, and that will be converted into gel on the anterior part. Now this particular gel, it will be coming back to the posterior end, and here we have the zone of solation. Now this particular phenomenon, it helps in the cyclotic movements. So as the amoeba moves towards the right side, so this helps in the movement of the cytoplasm also. So this is one of the main factor in cyclosis. So this solation and gelation is only because of the presence of the contractile proteins which are present in amoeba. So you have gone through the sol gel transformation theory in your BSc. So you must be knowing about solation and gelation and the presence of myosin and actin in amoeba. So let us take it as the first A factor which is affecting the cyclosis. The second factor can be the formation of pseudopodia. Now whenever the pseudopodium is formed on the anterior side of the amoeba, there is a huge rush of soul or the endoplasm. So again we are coming back to our amoeba. So if this is the amoeba and it is going towards the right side, so what will happen? The soul from the center of the amoeba, it will rush towards this end. So this is a soul, and when it rushes towards a particular end to form the pseudopodium, it will help in the cyclosis. So it will move the cytoplasm. So whenever the soul is thrusting onto a particular end, so what happens that at this particular place a vacuum is formed, so an empty space is formed. So what happens that soul from this side, it will enter, it will go to the central part. So with this particular movement of the cytoplasm, everything inside the amoeba will move. So that is cyclosis and that is circulation. So we can say that this cyclosis is the movement of the cytoplasm within the body of the amoeba. So this is the movement of cytoplasm. We can call it as cytoplasmic streaming also. Now the second factor which we are taking inside the circulation among protozoans is the movement of the food vacuoles. So we are talking about amoeba. So again we have to draw one amoeba, and suppose this is the amoeba. So you can make it of any shape because it is not having any definite shape. This is the nucleus, and suppose this is the food. This is the food, the red color food. So what will happen that the outer membrane of this amoeba, it will make a cup-like structure around the food. So that we call it as the food cup. So this is called as the food cup, and this red colored food will be at last will be taken inside this food cup. This membrane will be covered like this, and in the very center we are having now, this is the food vacuole. So This is the mechanism of the formation of food vacuole. So the process is called as, do you know what is this process called? Yes, this is called as phagocytosis. So here, phago, it refers to the solid thing, the solid food, and cytosis is the intake within the cell. So this is the taking in of the solid food. And if suppose this food is liquid, then we will be saying it as pinocytosis. So here the word pino, it refers to the liquid food. So suppose amoeba is taking in some oil droplet, so that will be pinocytosis. Now here in this case we are considering phagocytosis. Now food, red colored food, now it is in the food cup or the food vacuole. Now this food vacuole, it will move towards the center of the amoeba. So if we again make this food vacuole, now it is inside the amoeba and it is having the red colored food. Now this particular structure is the food vacuole. Now this food vacuole, it will move with the cytoplasmic streaming inside the body of the amoeba. It will be moving, and it will be moving due to cyclosis. So this food vacuole, when it moves inside the body of the amoeba, lysosomes, they are attaching to it. So what happens that food vacuoles, they are attacked by lysosomes. So actually lysosomes, they are containing the digestive enzymes. So suppose this is a food vacuole, it is containing a food, so this is a food, or we are making it as a red colored food. Suppose this is the food, and what happens, a lysosome will be attached to it. Suppose this is a lysosome, so a lysosome is coming and it is attaching itself to the food vacuole. Now what the lysosome is containing, so these are the digestive enzymes. So whenever this particular membrane is lysed, what happens that these enzymes, they are poured onto the food, so the digestion occurs. So in the due course of the cyclosis over here, when the cyclosis is going on, lysosomes keep on attaching to the food vacuole, and digestion process, it occurs inside the food vacuole, and the digested food is taken out of this food vacuole into the cytoplasm. This is called as absorption. So this is the process of absorption. So whenever this food is absorbed into the cytoplasm, what is left inside the food vacuole? Anybody? Yes, this is the waste. So I am again making a food vacuole. Suppose this is a food vacuole, and it is now containing only the waste materials. Suppose these are the waste materials. Now this body is called as the residual body. And this residual body, it moves inside the body of the amoeba. So it is again with cyclosis. It is moving, it keeps on moving, and when this body is only having the waste and every digestive material is taken out into the cytoplasm, it will move to the membrane and it will throw its waste products out of the amoeba. Now this residual body, it will move towards the membrane of the amoeba. Here it will be throwing out its waste products towards the outside. So this is called as the cell vomiting or exocytosis. So this process is called as exocytosis, or it is also called as cell vomiting. So this is the whole process of digestion, but here we are concerned with the movement of the food vacuole whenever it is formed in the amoeba. We are not concerned with the mechanism of food vacuole formation, but once the food vacuole is formed, how it moves inside the body of the amoeba, how it is getting connected with the lysosomes, how the digestion occurs, and how it is moving while the digestion is occurring, and the absorption process, and then the movement of the residual body, that is again with the cyclosis. So here in the heading we can add one thing, that this is the movement of the food vacuoles and residual body. This can be our heading. So I think you are clear, you are understanding this particular point. Students, now we are taking up the third point, which is the transport of gases in protozoans. Now here we are discussing about amoeba. So what happens is that this amoeba, it is living in fresh water. So here we have fresh water, and fresh water has low concentration of salts, and it has got dissolved oxygen in it. So we got dissolved oxygen in fresh water. It is present in quite a good amounts in fresh water. Now inside amoeba, the level of oxygen is low. So inside amoeba, again we have the dissolved oxygen. It is dissolved in the cytoplasm of the amoeba, but it is at low concentration. So what happens, that we have high concentration of oxygen outside. So here we have good concentration of oxygen. Inside we have got low concentration of oxygen. So what will happen, that oxygen from outside, it will be coming inside the amoeba simply by process called as diffusion. So oxygen enters in the same format. So this is very important to write, that oxygen is dissolved outside, oxygen is dissolved again in the inside of amoeba, but due to the concentration gradient is the difference between the concentration. So the oxygen is coming inside the amoeba simply by diffusion. Now if it is coming inside, now suppose we have an equal concentration here in case of amoeba. So we have dissolved oxygen which is coming from outside. Now amoeba is continuously taking this particular oxygen to different parts. So this is again with the cyclosis. Now this oxygen is used in different respiratory processes. In food respiration, it breaks down the food inside amoeba, and this is converted into carbon dioxide. Now again this carbon dioxide is in the dissolved phase. So here I am writing as D. So this carbon dioxide, it is at high concentrations. So this is at high concentration. And in the fresh water which is outside, we have the dissolved CO2 at low concentration. So what will happen, this particular carbon dioxide, it will be going out of the amoeba simply by diffusion. Again the process is the very same. This is diffusion again. So this is how the amoeba is respiring. It is taking in oxygen and it is giving out carbon dioxide. So the transport of gases is again with the help of cyclosis. Now next point is the transport of contractile vacuoles inside the body of amoeba. So have you gone through a word which is called as osmoregulation? So this is the regulation of water and salts. So if an animal is regulating water and salts, this is the process of osmoregulation. And the amoeba, it is regulating the water and salts with the help of structures which are called as contractile vacuoles. So it is regulating through the formation of contractile vacuoles. Now first of all we have to go by that why there is a need of osmoregulation in case of amoeba. So if we take back this amoeba on our right side, then what is the case? We should again consider that where this amoeba is living. So it is living inside fresh water. Now fresh water has got low salt concentration, and this is very important to know. Now inside the amoeba we have got high salt concentrations. Now we have gone through a particular phenomenon which is called as osmosis. Do you know what is osmosis? First of all, this is the movement of water. So this is not the movement of the salt. This is very, very important to know. So this is the movement of solvent, and here the solvent is water. So osmosis is the movement of water from the low concentrated solution to high concentrated solution. So that means the water from outside, it will enter into the amoeba. Have you gone through this? Are you understanding this? I am again trying to explain it, that the fresh water which is outside amoeba is low in concentration. We have got high concentration inside the amoeba. So osmosis is the movement of water. So what is the condition? That if low salts are present outside, so we got more of water outside. Low salts, high water amounts. But if we take the inside condition, we have got high salts but low water. So as the osmosis is the movement of water, so it will be from higher to lower amounts. So we have high water content, the high water amount in outside the amoeba, so it will be coming towards the inside. So this process is called as endosmosis. So it is endosmosis. Endo means it is coming inside the amoeba. If it is exosmosis, it is going out of a cell. So this is endosmosis. Now, due to this endosmosis, the water it is entering into the amoeba, and this is happening 24 hours 365 days. So water is coming inside amoeba. So what will happen if this much amount of water is coming inside continuously into the amoeba? What will happen to the amoeba then? So it will burst out. It will be bursting. So this amoeba will burst out. Now what the amoeba is doing for this water, for this incoming water, is that it has built a mechanism of osmoregulation, and this is with the formation of contractile vacuoles. So what happens? Small vesicles, they pinch off from the membrane of the amoeba, and these are called as contractile vesicles. So these are very, very small bodies which are pinched off of the plasma membrane of amoeba. So these are contractile vesicles, and these vesicles, they join together, they are joining together, and they form big contractile vacuoles. Now this is a contractile vacuole. Now what is the function of this contractile vacuole? That it is gathering water. This is the major function. So amoeba is having a large amount of water inside it, and this water will be gathered by this contractile vacuole, and as the water it enters into the contractile vacuole, it is bulging up. It is now bigger in size. Now when it becomes big in size, it will go to the membrane, it will attach itself to the membrane, and it will throw the water out of the body of the amoeba. So what is the contractile vacuole doing is that it is gathering water and it is throwing away of this water out of the body of the amoeba. So this is the maintenance of the amount of the water inside the body of the amoeba. So this is osmoregulation. This is osmoregulation. Now what happens when this contractile vacuole is gathering water? It is moving inside the body of the amoeba. It keeps on moving, and it is moving with the help of, again, what? It is cyclosis. So this is moving inside the body of the amoeba with cyclosis. It is gathering water, and finally when it bulges up, it moves to the membrane, it attaches itself, the membrane breaks off, and the water is expelled out of the body of the amoeba. So this is how the water is maintained, and this is the transportation of the contractile vacuoles inside the body of amoeba with the help of cytoplasmic streaming. So this is the circulation, this is transportation of food vacuoles, contractile vacuoles. So we are not concerned that how these contractile vacuoles are formed, but we are concerned that how these are transported inside the body of the amoeba. So this is our topic. So that was about the transportation of contractile vacuoles. Students, so I think you have understood the mechanisms of cyclosis. You have understood the mechanism of the movement of food vacuoles and the movement of residual body, the transport of gases, the transport of oxygen and carbon dioxide, the transport of contractile vacuoles in osmoregulation. And one thing you can add into this topic is it will be the transport of waste materials. So this is transport of waste. So these wastes, they are, suppose it is ammonia, so it is given out simply through diffusion, again from the cytoplasm with the cytoplasmic streaming in the form of ammonium ions. So we have a reaction which is, I think you are familiar with it, that ammonia, it reacts with water to form, it is forming ammonium ions NH4 plus and OH negative ions. So this particular ammonium ion which is formed, it is given out of the body of amoeba along with the water simply by diffusion. So this is a transport of waste products or the waste materials or the excretory products. So that was all about this particular topic, the circulation among protozoans. So here we had only talked about amoeba. In the coming lecture, we'll be talking about the paramecium. So it is again a part of circulation among protozoans. Thank you very much, and thanks for your patience.